Hey Bio 1 students, this is the pre-lab for the Osmosis lab. So if you are following along, this is going with lab 4 of your manual. We won't be able to do every aspect of lab 4 since this is a virtual lab, but we will be analyzing the data that you would have collected. So the first thing we would have done if we were meeting in person is you would be given a solution of 1 molar sucrose. That's table sugar like you see here. And we'd have to be careful not to make the lab all sticky. So 1 molar solution of sugar, and you would be asked to make 30 milliliters of each of these different concentrations. 0, 0.2, 0.4 molar, 0.6 molar, 0.8, and 1 molar. And you'd have to make, like I said, 30 mil. That would be this column here, V2. And you'd use the mass balance equation to figure out how much stock solution you needed and how much water you needed to add. I will assume that you know how to do that. You might want to work on this table if you're uncertain because you'll be quizzed on it a lot. Okay. You would also make 7 potato discs that are 5 millimeters long. Okay, so you'd cut these potato discs, and then you would take their mass, and you would write down their mass, like you see here, 23 grams, 21, 23, 20, 22. And then above them, you would write which of those solutions that you've made they're going to go into. So your 0, 0.2 molar, 0.4 molar, 0.6 molar, all the way up to 1 molar. And there's an extra one because you would also be given a beaker of solution labeled unknown sucrose concentration. So it's a concentration that you do not know. All right. So the next thing you would need to do is write a hypothesis about what you think is going to happen, the major trends of the data if you did this experiment. You don't have to predict what would happen at each concentration, but are the potatoes going to lose mass as the concentrations of sugar get heavier? Are they going to gain mass, and why? That's basically what you're looking for in your hypothesis. Typically, we write hypotheses in the if and then format. If is your explanation as to why the data is going to happen, followed by and, which is what you're going to do, and then you tell the reader what the data will do if your explanation is correct. If and then. For example, if stepping on the gas pedal of the car injects more fuel into the cylinders and I step on the gas pedal, then the car will go faster, right? So it's a hypothesis that's a predictive explanation. It's not just a prediction, it has an explanation built into it. So please try to do that in your hypothesis writing. All right, so let's get some background so we can write a good hypothesis. So if we have a beaker of water, and that water is above absolute zero, which it would be because it's above absolute zero on planet Earth, right? As long as it's above absolute zero, that means molecules are in motion. So if we have a beaker of water and we have molecules of dye in that water, they are in motion. They're moving around. It's hard to imagine that, but if you zoom in, even with a microscope, you can sometimes see this. The molecules of dye are moving. And in this case, we have a beaker of water where we also have a permeable or a semi-permeable membrane. So the gray hash marks are the membrane, and then there's pores in that membrane. So what will happen is these dye molecules are moving around, and they cross the membrane, and eventually they're equal on both sides. We go from a high concentration of dye to a lower concentration of dye. Here the dye is equally spread out on both sides of that membrane. Now for every one moving to the right, another one is just as likely to move to the left, maintaining that equilibrium. The molecules don't stop moving because they're in equilibrium. They're still moving. It's just that they're spread out and they're more or less equally concentrated on both sides of that membrane. Same thing would happen if you had more than one kind of dye. You have a few purple dye molecules on this side and more yellow on this side. Eventually they spread out and you have equal concentrations of the yellow dye on each side of the membrane, as well as fairly equal concentrations of the purple dye on each side of the membrane. But that's not really what we're measuring in this lab. What we're measuring in this lab experiment is not just basic diffusion, but osmosis. Osmosis is the diffusion of water across the membrane. So when would that happen? Because we didn't really see that here. We saw the solute, the dye, moving across the membrane. But what would happen if you made these pores smaller and the dye could no longer fit through those openings? That's what we have happening in this example. Here we have a U-tube, a U-shaped tube with a membrane, and this is a semi-permeable membrane that allows water to cross, but does not allow sugar to cross. Sugar's too big. And in this tube we have a low concentration of sugar over here, so a low solute concentration. And over here we have a high solute concentration, lots of sugar in that water over here. Now what happens is, if you set this up, you'll get this result where the volume of water changes, and this side increases in volume, this side decreases in volume. But what you end up getting is similar concentrations of water to sugar on each side of the membrane. So sugar to water ratios are about the same in this example here. So how could that happen? Well, if we zoom in and we look at water molecules, we will notice that they too are in motion. As long as there's any energy around, they are in motion. The water molecules on the left are free to move because there's not much sugar over here. They're not stuck to that sugar. If you've learned about how water dissolves things in lecture, you'll know that water forms hydration shells around other polar substances. So water is polar because it has polar covalent bonds between the hydrogen and the oxygen. That makes water partially, it makes the oxygen of water partially negative and the hydrogen partially positive. And so they will stick to their opposite charges. So sugar is a polar molecule, so water will stick to sugar. Now there's not much sugar on this side, so most of the water is free to move and it crosses the membrane. But the water on this side is heavily stuck to that sugar, forming hydration shells, which you may have learned that term in lecture. When water dissolves a substance, it's stuck to it, forming a hydration shell of water around that substance. This water is bound, it's stuck, it can't come through the membrane. This would be like you walking through a doorway, which you fit through just fine, but then you sit down on top of a horse or an elephant or something, and now you can't get back through the door. You don't fit anymore. So that's what's happening here. Water is moving from this side where it's free to move to this side where it's not as free to move. Water on this side is less likely to come back, so therefore the volume of water changes. So in our experiment this week, we're putting potatoes into different concentrations of sugar. Water in those concentrations can cross the membranes of the potato, but the sugar cannot. So you'll have to keep that in mind and think about what you might write down for your hypothesis. How do you think the mass of potatoes will be affected by the various concentrations of sucrose? Now, once again, you don't have to write about how each concentration will affect it, but what will the overall trends be as you go from 0 sucrose to 1 molar sucrose? How will potato mass overall be affected as you move along that gradient? Now, in living cells, we can see how they react and how different organisms react a little differently. And there's going to be some new terminology here: hypotonic, isotonic, and hypertonic. The C moved here. These words refer to how much solute, or the concentration of solute in a solution versus inside of a cell. Okay? Over here I talked about free water. Here I'm focusing more on the solute. So in this case, in this example, there's more solute over here than this side, right? So we're talking about which side of the cell membrane is there more solute, inside the membrane or in the liquid the cell is sitting in, okay? So let's say you plop an animal cell into pure water. That means there's more dissolved things inside the cell than outside the cell. That's said to be a hypotonic solution. That cell is sitting in a hypotonic solution. Hypo means less than. Like hypothermia is when you have a temperature less than normal. Hypotonic means the cell is sitting in pure water, less than normal, or less than what's inside the cell. So when that happens, the water outside the cell isn't really stuck to anything. There's no sugar out there, no salt out there, whatever. That water can rush into the cell faster than it can leave the cell. And when you do this to an animal cell like this red blood cell, the cell lyses or bursts. It swells up and pops. When you do this to a plant cell, they're perfectly happy. They swell up, but they don't burst because they have a cell wall. And this is actually how plants get to be stiff and robust and stand upright by having that turgor pressure inside. All right, my phone rang and I was interrupted. So plant cells fill up with water. They become turgid. This is how a plant stands upright. Not all plants, but like celery, for example. Think of celery that's good and healthy. It's crunchy. Its cells are full of water. They're turgid. That makes celery nice and stiff. But if celery is wilted, it's floppy and it's no good. And that's what you have in this middle situation. So if you place an animal cell in an isotonic situation, that means there are equal concentrations of solute inside the cell and outside. That means water can come in and out at the same rate. Animal cells love this. They're happy. They're good to go. Plant cells actually don't like this as much. It doesn't make them as stiff. They're not as turgid. They are flaccid then. And this is when you start getting wilting in a plant because it can't stand up as straight because it relies on this pressure as kind of its skeleton essentially to stand upright. In the case of like a lot of herbaceous plants like grasses and other herbs. Now, in the case of a hypertonic solution, that's when you place, let's say, an animal cell into something like salt water or sugar water, something really salty or really sugary. That means the water outside the cell is stuck to those substances. There's more free water in the cell than out of the cell, so water can leave the cell more easily than it can come in. When that happens, the animal cell shrivels up. Plant cells also have an issue with this where they dehydrate, water leaves the cell, the plasma membrane shrivels up inside of that cell wall, and this is said to be plasmolyzed. All right, so you have the background you need now for a hypothesis. So what are we going to do? We are going to make a graph then. So what would have happened in class is you would weigh the potatoes, or take the mass of the potatoes, before they go into solution and after they go into solution. Okay? And you would look at how much they gained or lost mass. And what you're going to do with that information is make a standard curve. Now, this is an example standard curve here. It has nothing to do with our data. But a standard curve is essentially a scatter plot that you would make in Excel. And standard really refers to known concentrations. You have standards. You have the sugar solutions that you knew what concentrations they were. Okay? And then you make a graph of those standards, and then you can figure out what the concentration of your unknown was using your graph. So using the best fit line here, you can determine what the unknown concentration was. So here's an example of some potato data. So let's say your data looked like this. It might not look like this. This is just an example. Here we have sucrose concentration, and we have change in potato mass in grams. So this is how we're going to graph the data. Your manual on number 6 tells you to convert this to percent change. I'm not going to have you do that this semester with this online lab. So we're just going to graph change in mass, okay? All right, so here we have how much mass the potatoes gained or lost at those different sucrose concentrations. You can see that here, and there's a best fit line here. So what you would want to do is you would want to also insert a equation of the line right here so that you can figure out what the unknown concentrations were. And you'll write a figure caption just like you would do for any graph. I can't guarantee how good this one is, but there's a figure caption there. So that, in this case, by the way, I want to point this out. In this example, sample size is 1. In your case, sample size will probably be 6. So you'll have error bars and other things on your graph that you do not see on this example. So there are a couple of questions that are in your manual, number 10 and 11, that you'll need to answer in your manual, okay? Or you'll need to answer on your assignment. And those include, well, 10 and 11, and then there'll be some additional questions I may ask on the worksheet. But the two questions are, what concentration is isotonic to the potato, and what is the concentration of the unknown solution? And the way you figure that out, there are two different ways. One way is simple but a little less accurate. So let's say, for example, that your potato let's just say that we want to do this one here. What concentration is isotonic? What should a potato mass do in an isotonic solution? Well, it shouldn't change at all. It should be zero change in mass. So what you would do is you would find where zero and the best fit line cross. And that doesn't mean there will be a dot there. I'm not asking you which concentration that you tested is close to isotonic. It's what actual concentration, if you had tested every possible concentration here, would be isotonic. So you can figure that out here, or the more scientific way to do it is, if you remember, the equation of the line is y equals mx plus b. I'm asking you what concentration would be isotonic, so what concentration would cause no change in mass. So that would be, you'd have to reformulate the equation to x equals y minus b divided by m, right? Y is zero change in mass. So you substitute 0 for y and you solve for x, and you could get a pretty accurate answer there. You can do a similar thing with the concentration of the unknown solution. You'll look at the average change in the potato mass for the unknown, and then you'll solve for x using the equation of the line, and that will tell you your estimate of the concentration of the unknown solution. So when you spit the equation out using Excel, it'll give you y equals mx plus b, and so you can solve for y or solve for x depending on what you're trying to answer. And if you're confused which one's x and which one's y, remember, if it's x, it has to do with what's on the x-axis, and y has to do with what is on the y-axis of your graph. One other thing I want to point out when you're interpreting your data: if your data ends up looking like this, bear in mind that here's 0. This is no change in mass. If you have some solutions that fall above no change in mass, that means they gained mass. They got heavier. And if you have potatoes that are below 0, that means they lost mass. They got lighter, right? They lost mass. Water left the potato. And sometimes students just say, hey, potatoes lost mass in all of my data. But that's not true if you have some of these data points that are above 0. So keep an eye on that when you're working on explaining and interpreting your data. All right? So that does it for this video. You'll see some data on the website, and you'll see a worksheet to fill out. So do that to the best of your ability, and don't forget to reach out to your lab instructors if you have questions. Good luck.